As we mentioned earlier, the main purpose of neuromodulation is to broaden the repertoire of synaptic transmission and neuronal function over many different timescales, and potentially over a wide range of targets throughout the brain. Most of the classical neurotransmitters we've talked about so far are so-called small molecules. This makes a certain amount of sense that you might want to have neurotransmitters be small, since smaller molecules tend to diffuse faster. But not all neuromodulators are small molecules. Another interesting class of neuromodulators are the neuropeptides. Neuropeptides are proteins, usually 5 to 20 amino acids in length, small as proteins go, but many times larger than classical neurotransmitters. They're often derived from larger precursor proteins that are produced in the cell body and then transported to the nerve terminal and cut up into smaller pieces. In contrast to classical neurotransmitters, neuropeptides act exclusively through GPCRs. Interestingly, in many cases, neuropeptides are released along with classical neurotransmitters at synapses, and they act as neuromodulators. Thus far, over 100 neuropeptide varieties have been identified, and the list is growing year after year as more are identified. Examples of neuropeptides include hypocretin, which is used to regulate sleep, leptin, which is involved in appetite, and substance P, which is known to modulate pain sensation. There are many, many more. To highlight the mechanisms of action of neuropeptides, let's look at a particularly interesting class of them, the opioids. The activation of opioid receptors modulate pain perception, and their discovery is a good example of how decades and centuries of observation can come together and teach us about how the nervous system works. For centuries, it was known that opium, derived from the poppy flower, has an analgesic effect. That is, it reduces our perception of pain. Opium was probably the first robust painkiller known to mankind. It wasn't until the 19th century that chemistry advanced to the point that the components of opium could be isolated. Opium is a cocktail of alkaloid chemicals, but notable within the cocktail are morphine, which makes up about 1/10 of opium, and codeine, which makes up a few percent. Scientists noticed that these compounds also had analgesic properties, though the mechanisms were still poorly understood. In the late 19th century, a synthetic opioid, heroin, was first synthesized. It's basically morphine with a few extra methyl groups tacked on. Like many drugs, both therapeutic and recreational, the creation of a substance that is metabolized or processed by our bodies into some kind of neuromodulator is a pretty common theme. A variety of synthetic opioids have been created, and you may have heard of some of them. Hydrocodone, oxycodone, and OxyContin all belong to the same family. All of these drugs are important tools in medicine's arsenal, but all are also increasingly targets of abuse. Back in the 19th century and early 20th century, there was an increasing understanding that opioids were a family of substances with common effects on human physiology and common structure. But at this point in history, it was as if the keys were known, but the lock was still mysterious. It was clear that substances existed that had a powerful effect on our perception of pain, but it wasn't clear how these substances had this effect. It wasn't until the 1960s and 70s that the picture started coming into focus. Evidence for opioid receptors started to emerge in the 1960s, and in 1973, Pert and Snyder identified the first definitive characterization of an opioid receptor and its binding site. The receptor that they studied would eventually come to be known as the mu-opioid receptor. Eventually, 4 different kinds of opioid receptors would be identified, dubbed delta, mu, kappa, and nociceptin. Why does the body have these receptors that bind these exogenous substances like morphine in the first place? Well, perhaps not surprisingly, they didn't really evolve to bind morphine at all, or at least that's not their primary function. It turns out that there are a whole host of endogenous opioids that the body produces naturally, that are the natural triggers for the opioid receptors. Among these are enkephalins, endorphin, and dynorphin, each targeting a different receptor class. Interestingly, while exogenous opiate substances like morphine are small molecules, the endogenous opioids, those found naturally in the body, are all neuropeptides, small protein fragments. The opioid receptors themselves turn out to be GPCRs, And as you might imagine, developing substances that can effectively target these receptors is an area of great medical interest. It might seem strange that a small exogenous molecule like the opiate morphine can activate the same receptors that are endogenously activated by much larger neuropeptides. But this is actually pretty common in neurobiology. Just like a real-life lock and key, only particular surfaces on the key are critical to opening the lock. As a result, you can open up a lock with its intended key, but you can also use a lockpick to trick the lock by simulating the distinctive surface of the key, even briefly. In other cases, it's possible to exploit some other part of the lock mechanism to defeat the lock. Likewise, some exogenous neuroactive substances bind to receptors, but not in their usual binding site. It's not entirely clear why the poppy produces alkaloids that are so effective at binding the opioid receptors, but it's probably not an accident. Plants synthesize all kinds of neuroactive substances. Think tobacco and nicotine, or THC and marijuana plants. A good part of this probably has to do with defense. A plant can't really defend itself beyond making those who eat it sick or dead, either killing the would-be consumer or teaching them a lesson. Substances like nicotine are a drug of abuse in humans, but it's deadly for insects who eat the plant. Likewise, neuroactive effects of plant-based opioids likely play a defensive role, making their targets drowsy or nauseous. Nature is in many ways characterized by a grim evolutionary arms race, producing poisons and toxins that elegantly co-opt and usurp the molecular machinery of our bodies. However, as terrible as some of these poisons are in nature, they're a boon for neuroscience, providing essential clues to the function and structure of the molecular machinery of the nervous system. To the extent that we can understand and harness nature's creations for therapeutic purposes, we benefit greatly from the comparative bounty of neuroactive substances that nature has wrought.